Welcome back, everyone. Next up, we have Acurx Pharmaceuticals, trades on the NASDAQ under the symbol ACXP, and is a clinical stage biopharmaceutical company focused on developing an entirely new class of antibiotics for difficult to treat infections. Please welcome its president and CEO, David Luci. Welcome, David. Nice to see you.
Well, thank you, Anna. Thanks for having me back.
Nice to have you back as well. So the floor is yours, and call me back when you're ready for questions.
Excellent. Thank you so much. Good morning. My name is David Luci, President and CEO of Acurx Pharmaceuticals. We are developing a new class of antibiotics, first new class in over 30 years, if FDA approved. And we have polymerase 3C inhibitors. And we'll tell you about the mechanism of action of our lead antibiotic candidate in trials now to treat C. difficile infection. So we started our company in early 2018 by acquiring our lead antibiotic candidate from two retiring professors from University of Massachusetts, where our lead antibiotic candidate was discovered. Its name is ibezapolstat. Its mechanism of action is to kill C. difficile bacteria by disrupting the polymerase 3C enzyme, which is the primary catalyst for making new DNA for gram-positive bacterial cells, including C. difficile. So C. difficile, which is the clinical target of our ibezapolstat, is an unmet medical need. The CDC suggests there's an urgent threat, an urgent need for new classes of antibiotics to treat C. difficile. There are 3 antibiotics currently used for mild to moderate C. difficile, and 2 of the 3 are extremely old, decades old, 50, 60, 70 years. And one, fidaxomicin, is also a little bit long in the tooth. It's coming off patent in about 3 years. It's a significant market, which we'll talk about, $1.7 billion market. And because the lead antibiotic candidate in the market, oral vancomycin, is so weak, it is a vulnerable target for picking off frontline therapy. So we have phase 2B clinical trials ongoing, a clinical trial ongoing, and we expect to take an interim look at our data in C. diff patients when we get to 36 patients later this year. Our phase 2A trial data was lights out good, 100% cures with no reinfections 30 days after the end of treatment. On the basis of that data, we IPO'd in the middle of 21, and we've now done 2 registered direct offerings since then, including one just a couple of months ago on May 18th. So what we're seeing in our phase 2B is slow enrollment, which is consistent with several publications and other company experiences. The conduct of clinical trials has slowed significantly since the behavioral patterns of people have changed due to COVID. But our data is lights out, and we've taken steps to address the slow enrollment by taking our clinical trial sites from 12, which we started with, to 28 trial sites, which we have right now. Now, we have a dual impact, or a second dimension of use of our ibezapolstat. While we're curing the direct C. diff infection, we're also restoring the microbiome, fully restoring the microbiome by day 3 of a 10-day treatment regimen, which is highly unusual in our experience for an antibiotic to actually restore the microbiome instead of disrupting or destroying the microbiome. We also have favorable bile acid ratio. So when you have significantly more secondary to primary bile acids, that's a scientific marker for a low risk of reinfection, which is what we've shown thus far. So we expect that trend to continue because of the scientific underpinnings for why we've had no reinfections. So we established an independent data monitoring committee, which is organized to take an interim look at our phase 2b trial data when we get to 36 patients enrolled. And when they do that, they will give us a recommendation on whether to continue enrolling or alternatively recommend to early terminate the 2b trial and move on to phase 3, much like our scientific advisory board had done when they recommended early termination of our 2a study. This chart shows the recent clinical trials in the C. difficile indication. Vancomycin, oral vancomycin is the standard of care. Fidaxomicin, which has a few years left on patent, a Merck product, is prohibitively expensive, but clinically a little bit better than oral vancomycin because of the pricing and because it's the new antibiotic of the two. It's used last line, but still does quite nicely revenue-wise. Ridinolazole from Summit Therapeutics failed phase 3, and that drug is now finished. So we're looking at 100% cures and 100% sustained cures, which is a fancy way to say no reinfections, based on a small number of patients, 10 out of 10 in our 2A trial, but with scientific underpinnings for why that data should hold. And we expect that it will hold. So antibiotics are still the gold standard to treat mild to moderate C. difficile. There are other things that have been tried, like fecal transplants in severe cases. Vaccines have been tried, most recently by Pfizer, before them by Sanofi. Both failed phase 3. So Summit, as I mentioned, failed with ridinolazole. And so the seas have kind of parted for us with our data and our price point that we can expect due to a very cheap manufacturing method. We expect frontline therapy to be a real opportunity for us. So our pipeline behind ibezapolstat right now is ACX375C, which is at the lead optimization stage of development. And we expect that process to be completed in the second half of this year. And then we'll move on to IND-enabling pre-clin tox work with another oral antibiotic Pol-3C inhibitor. It too will be a new chemical entity, and it too will qualify, and we would apply for qualified infectious disease product designation from FDA and fast track designation from FDA, just like we've already obtained from FDA for ibezapolstat. We're also working with Leiden University Medical Center and the Dutch government on building our pipeline of Pol-3C inhibitors behind ACX375. And we hope to have 2 to 4 more products in that pipeline over the course of the next couple of years. ACX375C also targets the Pol-3C enzyme, but in patients with MRSA infections. So the first clinical target for that would be abscissae, skin and skin structure infections. So we talked about the mechanism of action. We're able to kill the C. diff bacteria by disrupting the activities of the Pol-3C enzyme, which is the primary catalyst to make new DNA for new C. diff cells or new MRSA cells. Interestingly, there is no Pol-3C enzyme in healthy cells. So our class of antibiotics leaves healthy cells alone. So we mentioned the market size, 1.7 billion. You can imagine a frontline therapy in this market would be quite attractive. And given the frontline therapy is vancomycin, you see it's very vulnerable because oral vancomycin has a decent cure rate, but then approximately 30% of the patients reinfect. And the patients that reinfect, 70% of the time, have chronic reinfections. So we think that's why Pfizer and Sanofi tried to develop vaccines to be frontline in C. difficile. We think one of the main reasons is that the standard of care, oral vancomycin, is vulnerable to something being better. So in the 2A, we were able to establish 100% cures with no reinfections. Our drug was well tolerated with no serious adverse events. We were able to see fully restored microbiome in the process, just 3 days into a 10-day treatment regimen. The IP and regulatory protection is substantial. It's 10 years, and it's not 10 years from today. It's 10 years from whenever we get FDA approval. So having sold to Big Pharma in the past on a 7-year DCF model, that's a pretty standard way to go. So we're definitely going to get our value out of the drug in our anticipated M&A, which we will proceed with after the 2B data reads out. So how is it that our ibezapolstat works so effectively at treating C. difficile and not disrupting the good bacteria in the gut, known as the microbiome? The reason is because we're a very narrow spectrum antibiotic. So we're able to kill C. difficile bacteria, which you can see on this chart is Firmicutes. It's within the Firmicutes subclass. But we're not able to kill any other bacteria. Unlike vancomycin, which is more broad spectrum and so is disruptive of the good bacteria in the gut while it's killing the C. diff bacteria. This chart shows the major subclasses of bacteria in a healthy gut or a healthy microbiome. And the only thing that we touch is the Firmicutes, making it an ideal candidate to treat C. difficile patients. So our plan is to complete the phase 2B trial, get the data out there in a press release, meet the FDA as soon as we can thereafter to design and negotiate the phase 3 trial design. While we're doing that, we will be exploring strategic alternatives. And, we've already soft launched that, and we're going to hard launch it more formally after the data is in our hands. And if we're not sold prior to starting enrolling phase 3, we're going to park one of the 2 phase 3 trials that FDA is likely to ask us for. And the other phase 3 trial, we're going to fund the company to get to an interim look on the first of the 2 phase 3s. So if we're unable to sell the company prior to getting to the interim look on the first phase 3, we'll redouble our efforts thereafter with even more positive data in hand in our expectation. But why do we feel so strongly that the phase 2B will be successful? And the reason is we're getting over 100 times the concentration of our medicine into the site of the C. difficile infection in the colon than is needed to kill the C. diff. So that's like killing a bug with a bazooka. It's just a real lot of concentration. In addition to that, the fact that we're restoring the microbiome while the patients are just 3 days into a 10-day treatment regimen means that those patients are going to be a very low risk of reinfection. And that's the secondary endpoint. How many reinfections are there? So there are scientific reasons why we see this as a high probability of success. ACX375C is still preclinical at lead optimization, so I won't dwell on this much other than to mention we're almost done with this process and selecting a lead. And we do have a significant target with MRSA infections, which are still 52% of all hospital-based infections in the U.S. And we have an open application to CARB-X to fund $11.3 million of a $16 million project, which would take 375 straight through to the start of phase 2 over a 5-year period of time. and we will announce that we're told we'll find out from Carbex no later than October. So the biggest value driver outside of our company and our trials is the Pasteur Act, which is deeply in the legislative process and has bipartisan support through our affiliation with the Antimicrobial Working Group. We're confident this will be passed, but it's Washington, and the timing of when it will be passed is completely up in the air. So we're not counting on it, but if it is passed, it would have a dramatic impact on our company by paying for our phase 3 trials, as well as providing at least $375 million to the company, and we would then stockpile public health facilities with our drug under a license agreement with HHS BARDA. So we have an experienced management team. I come at it from the corporate side with deep experience in mergers and acquisitions, financings, from the legal side and the accounting side, operationally. Bob DeLucia, our executive chairman, runs our R&D program and has deep product development experience starting at Pfizer in 1972. Our management team does include 2 folks that were instrumental R&D folks that took Cubicin out as an antibiotic for Acurx Pharmaceuticals before it got acquired by Merck. And since our 375 drug is so similar to Cubicin, we figured they would be good ads to run our 375 program. That is Shang Yu and Larry Morton. Our board of directors includes Joe Scuderi, the former chairman of the pharmaceutical division of Johnson & Johnson, and Jack Dean, who led preclinical development at Sanofi for years. Our scientific and corporate advisors include Fred Hassan, who has been very helpful with our business development activities. Fred was the chairman and CEO of Shearing Plow before it got sold to Merck. Our recent developments we've discussed. Our near-term catalysts include the decision by Carbex on the $11.3 million of funding, which is close to $1 a share for us, along with our phase 2B data and our lead optimization, completion of our lead optimization with ACX 375. So our exit strategy has 2 facets. When we get through the phase 2B trial, plan A would be to sell the company to a big pharma player in the antibiotic space. And while we're doing that, we're going to continue to prepare for phase 3 as if the M&A is not going to happen. And we'll see which we're able to do. And if we're enrolling phase 3, for lack of terms with big pharma that our board accepts, we will try again when we have the interim look on the first phase 3 trial. And that's Acurx Pharmaceuticals. We have 10 years market exclusivity, 5 for being QIDP and 5 for being new molecular entity or new chemical entity for ibezapolstat, our lead antibiotic candidate. And we expect the same 10 years for our second antibiotic candidate, and that's alongside of the patents that we have. Anna, if you'd like, I'm happy to answer any questions.
Great job, David. Thank you so much. And congratulations for all these updates. You mentioned a lot about the phase 2B clinical trial. So let's talk a little bit more about that. How is the blinded pooled data looking so far in the phase 2B clinical trial you mentioned?
The data is looking excellent. It's the only way I can describe it. It's a blinded trial, a double-blind trial, but the data is pooled, and we're able to see cures versus non-cures and who's getting reinfected versus not getting reinfected. And we're also able to look at vancomycin data, which is chock full. There's been many, many clinical trials with vancomycin over a 60-year period of time. So you could kind of get a real good sense for what vanco is going to do. And, we have almost, no, non-cures. I mean, almost everyone's cured. So we have a real good feeling about the outcome. We announced 26 patients enrolled in the last earnings call on May 14th or so. And we've continued to enroll since then. So we're going to come out with a hard number in our next earnings call on August 14th. But we're in the single digits. We're delighted with that.
And when do you expect to complete enrollment?
We expect to complete enrollment in the second half.
And let's talk a little bit about CarbX. When do you expect to hear from CarbX about funding your second antibiotic program for a 5-year development project?
We're told by CarbX, quote unquote, no later than October, we'll have a decision from them.
And so what would the passage of the Pasteur Act do for your company? And maybe explain what the Pasteur Act to our viewers.
Sure. The Pasteur Act would be a dramatic sea change for our company and for any other antibiotic company, incubator company like ours, which has a new class of antibiotics, number one. And number two, that new class of antibiotics has to treat a serious or life-threatening infection. Now, there are not that many out there since no new classes have been approved by the FDA in over 30 years. So the Pasteur Act would be dramatically impactful for us because it would pay for phase 3, and it would mean, a minimum. They would do a pharmacoeconomic buildup of the value to public health of having ribezapolstat available at market, and they would then allocate us at least $375 million over 10 years, or up to $1.5 billion over 10 years. So I'll exclude the 1.5 and just focus on the 375. That's 37. It's straight line, $37.5 million a year for 10 years to stockpile public health facilities. For a company right now that's a $30 million market cap company, and you could ask yourself, if Pasteur is passed, how do we know that we'll be designated under Pasteur to receive that money? And I think the answer to that is, how many, it's rare as hen's teeth to have a new class of antibiotics, to have one that we already know has been designated by FDA under the QIDP designation as treating serious and life-threatening infections. We would seem to be straight down the middle of what they're looking to do. I think the legislative intent behind the Pasteur Act, which has bipartisan support, is to do like they do in England, which is, instead of a push incentive like these small NIH grants that incubator companies get during the R&D period, let's have what they call a pull incentive. Scott Gottlieb talks about this a lot, the former head of the FDA, and they have this in England, where they say to young, struggling incubator companies like Acurx, hey, if you guys can find a way to get your antibiotic through the R&D gauntlet and to the market, there's a carrot out here for you. And that carrot is the value to the public health system of having your drug at market, meaning there's going to be less people sick and hospitalized with C. diff, less people getting other antibiotics that are less effective than ours. And there's a cost to all of that. And, by having your drug at market, we'll reimburse you for what we see as the benefit of your drug when it gets to market. So you won't have the problems that have plagued the antibiotic sector for the past 20 years. So it would dramatically change and incentivize and cause the antibiotic product pipeline to vastly expand, which is what we need because of antimicrobial resistance. Bacteria is just finding ways easier and easier to get around being killed infections continue because antibiotics are overused and the bacteria recognizes the medicine as medicine.
Well, thank you for clarifying that. And let's talk a little bit about the stock price. What are some near-term value inflection points other than the phase 2b data could drive up the stock price?
Well, there's going to be 2, if not 3, poster presentations we're doing in the second half at scientific conferences like ID Week. And they have additional data outside of the 2B trial that have kind of been percolating, including data on how well does ibezapolstat do killing bacteria that's fidaxomicin resistant or vancomycin resistant, metronidazole resistant. And we're finding great results even among patients that have resistance to 1, 2, or all 3 of those other possible candidates to treat C. diff. We're actually effective, whereas they're not. So there's those 3 poster presentations that will be coming out within the second half, along with a decision on the CARB-X funding, lead optimization. When we file for QIDP designation for ACX 375, that will be news. And as you mentioned, the 2B data itself will be news.
And David, if the company is not sold within the next 5 years, where do you see the company in terms of product pipeline?
Well, as you see, based on where we are with the University of Leiden, we would expect to have excavated 2 to 4 additional PAL3C inhibitors. And 5 years from now, we would expect our lead to be at market treating patients with C. diff. PASTEUR Act will be passed. It's a question of when, not if, which would really fortify our company. We would probably be up to 100 to 150 employees, most of them in sales and marketing. Our second drug, ACX 375C for abscissa infections, MRSA infections, would be in phase 2 clinical trials. And we would have 2 to 4 preclinical products behind that, all of which are PAL3C inhibitors.
Well, continue this great work, and we appreciate you coming on the conference again. And please join us again in the future with some more updates.
Definitely will do. Thank you, Anna.
Thank you, David. All right, everyone, stay with us. We'll be right back with our next presenter.